Mom, I didn't say Eugenia. Guess what? I said this one.
Them are the two you have to look at between these two.
Oh.
The L word and the last one.
And start with you.
Oh, shoot.
All right. Everybody can hear me and can see the slides? Okie dokie. All right, so we got a number of slides to get through. I've changed some things around, so don't freak out when the slides you downloaded last week might look a little bit different from the slides this week. I've also tried to attach my old mouse so it doesn't have such a hair trigger on it compared to the super duper new mouse that came with my new computer. All right, so last we left off, we were just starting to get into all this hard stuff about the prokaryotes. Prokaryotes, almost just another fancy name for bacteria. All right, and we talked last time about how when the baby comes through the birth canal, baby is born with a whole sterile intestinal tract, and baby is going to pick up the bacteria from mom's birth canal, from mom's vagina, usually starting to colonize itself with the typical bacteria like lactobacillus that colonize the vaginal tract or the birth canal. Babies who are born by C-section actually get colonized with bacteria that's typical of the skin or the microbiome of the skin, like Staph epidermidis. So I find that kind of interesting. And, you know, as a general rule, you know, and you'd hate to say this stuff because it tends to make moms who have to have C-sections feel a little bit guilty, but, you know, C-sections happen for a reason. And I think as a general rule, babies that are born vaginally have a healthier, you know, first year of life than the babies who are born by C-section, but they tend to catch up. And some of this is thought to do with that microbiome. All right, so now we're getting in the hard stuff that I said, don't get obsessed too much about all this differentiating that the microbiologists like to do of getting into our, you know, our kingdoms, our phyla, etc., and obsessing over what gets classified as alpha proteobacteria versus, you know, gamma proteobacteria. We will end up sorting out a lot of our bacteria and discussing them in the end by what system they invade. That's how they present themselves to us as medically important. But we're going to go through the classifications because we're using a textbook that does use those classifications, so we'll go through it. All right, and I think I just did go back here a little bit. All right, so like we said last time, we're going to classify bacteria, and we do this medically, largely by their shape and staining pattern. Gram-positive. Is it gram-positive, you know, cocci, or is it a gram-positive rod? Is it a spirochete? Is it gram-negative? Or is it acid-fast? These are going to be the way we pretty much delineate our bacteria. You need to know that Berge's manual is the definitive classification where the microbiologists get together every year or two and they reclassify and shuffle things around based on the newest research. A lot of this is coming down to what they see as DNA evolutionary connections between bacteria. So sometimes you'll find a bacteria that was once classified by one name will now get classified by a new name, and it makes it more confusing. But that's the way things are going. Everything is getting down to the nucleic acid level for classifying bacteria, nucleic acid for comparing our evolution, treating things down to the nucleic acid level. Our vaccinations are now down to the nucleic acid level with our new RNA, you know, vaccines. So this is technology evolving in real time for all of us. All right, so a lot of this, when I say this classification by nucleic acid level, I talked last time about the infamous C. diff bacteria was always established to be with the Clostridium group of bacteria. Nucleic acid studies ended up, deciding that it really needs to be in a whole different genera. And so Clostridium was reclassified in 2016 as Clostridioides. It's harder to say, but they kept it, with a name so you could still refer to it by C. diff because there was no taking this away from the common parlance used throughout medical establishments. And so we can still refer to the same organism as C. diff. It's still going to be anaerobic, generally gram-positive, spore forming, and we're going to take all the same precautions of constantly washing our hands and eradicating those spores from floating around the ICU so that you don't get C. diff diarrhea. I've had it when I worked in the ICU and it's floating around everywhere. I can tell you it's no fun. I was not immunocompromised, so I got over it in about 5 days. But the day that I got diagnosed with it, I almost passed out when I was on rounds in the ICU, just like, whew. So I can tell you, not fun stuff to have. All right, so taxonomy, we use the classification systems, the phonetic going on all those physical types, shape, gram staining, physical appearances, or phylogenetic, which is really getting down to that nucleic acid level. And genotypic kind of is the same as in comparing what genes each bug carries. Polyphasic is really pretty much what is in use, is combining all three of those. So there isn't really a widely accepted species definition. And it's kind of getting, like I said, more and more that they hybridize DNA probes to the microbes and try to see if there are preserved identity sequences in which they can kind of use to classify the species. All right, so this is where I kind of started switching around the slides because I realized that maybe some things didn't make as much sense as they could have. So let me go back here. All right, most of our bacteria we're going to classify based on the world of gram negative and gram positive, the two big cell wall archetypes, all right? So peptidoglycan is only found in our bacterial cell wall. We'll see, that's why I'll keep repeating this, is we'll see our big antibiotics like penicillin, vancomycin, target the making of the bacterial cell wall because it is so unique to the bacteria. Our cells don't have it. Now, peptidoglycan, also known as murine, is really thick in gram positives, thin in gram negatives. And in gram negatives, that thin layer of peptidoglycan will be sandwiched between an inner and an outer membrane. I know I repeat this a lot, but we forget this a lot. The overall function of our cell wall is going to confer strength and give that rigid shape, whether you're a coccus or whether you're a bacillus or whether you're a spirochete, to the bacterial cell. It maintains the shape and prevents lysis. That cell wall really doesn't let a lot of substances enter into the bacteria. And we'll find that the bacteria, and we're not going to go through this real in depth here, and I took out some stuff about the porins and all this, is that the bacteria will have a lot of the same functions, kind of like our plasma membrane does, only they'll have it in the cell wall where it really restricts what can get inside of the cell wall, what nutrients it needs to get in there. And so there will be kind of a chaperone system, some sort of insertion of pores and channels that'll allow only certain substances into the cell wall. This is all to protect that bacteria from osmotic lysis. So water, obviously necessary for life, is going to move freely across both the cell membrane and the cell wall. The peptidoglycan is relatively porous and does allow water to enter in. But of course, at some level, it has to restrict it too, so that the cell just doesn't burst. And there's a lot of research now going on into the movement of materials inside of and outside of the bacterial cell wall. That's why I'm kind of limiting some of this stuff, because it's changing. All right, so in your gram-positive cells, the ones with lots of peptidoglycan, most substances are going to be able to move in through the cell wall with little difficulty. Big nutrients are going to require exoenzymes. These are going to be extracellular enzymes that are made within the bacterial cell cytoplasm, but they're secreted outside of the cell membrane, and they function at the cell wall to help bring and break down large macromolecules into smaller components so they can allow entry through the cell wall and then eventually into the cell membrane. Since the cell wall is so porous, secreted proteins from the bacteria are able to be secreted outside of the plasma lemma and out to the outer cell, out to the cell wall, to allow that level of regulation. Okay.
Does that mean that they don't do endocytosis? They don't.
Oh, they'll do endocytosis, but
Okay.
But it's going to be very, very regulated and controlled as to what they bring in. Okay. All right. Now, gram negative is a little bit more complicated because of that sandwich layer. Okay, so we have more ingredients to deal with. So the outer membrane, and we're going to go from the outer membrane of the cell wall and work ourselves in. So the outer membrane is the big thing that's going to distinguish the gram-negative bacteria. The gram-positive don't have this. The outer membrane is a lipid bilayer, but it's not a phospholipid bilayer. It's not the same as the plasma membrane. It contains some phospholipids, but this is more confined to the inner leaflet part of this outer membrane. The outer membrane is mostly glycolipids, and the big glycolipid that's out there is lipopolysaccharide. And lipopolysaccharide is what you always have to think of synonymously with endotoxin. So this is what's going to be responsible for endotoxic shock. Endotoxic shock starts out with septicemia, blood poisoning, right, with bacteria that leads to endotoxic shock from gram-negative organisms releasing this lipopolysaccharide, specifically the lipid A component. The human immune system is highly sensitized to lipid A, okay? It will start a whole cascade of immune events when it encounters lipid A or lipopolysaccharide. And this is preserved across sort of all the evolutionary species that have lipopolysaccharide, showing that like this is a surefire indicator of infection taking place, and our innate immune system responds to this promptly. We don't even have to start with making antibodies and going to our adaptive immune response. We're going to start with our innate immune system. So this is ancient. All right. So the only known function of having the outer membrane is protection. And gram-negative infections, because of this protective outer layer, are almost always more difficult for us to treat. This is why you'll see when it comes back gram negatives, we're all like, ooh, gram negatives. Okay, we know we're dealing with gram negatives. This is going to be a little bit harder for us to mount our antibiotics and eradicate this infection. And lipopolysaccharide is the critical barrier protective key here and the big thing that we're fighting. And when we start fighting it and we go from septicemia, sort of infection in the blood, to actual septic shock, this happens as a result from the interplay between the gram-negative microbes and our big immune response. It's like nuclear war between the two sides, okay? So no détente, we're fighting each other here. And we throw everything at it with our immune system. We're gonna start all that innate immune response stuff of putting up our temperature, getting fever. Your heart's gonna start pumping fast, you're breathing fast, all these kind of big signs of inflammation. And then it goes on down the road to affecting all of our organs where we get hypotensive. Our blood pressure goes through the floor as our heart rate is going way, way up through the roof. We become hypoxic. We're not getting enough oxygen into our tissues. We start shutting down our kidneys and our whole urinary system. So you go from, oliguria, intermittent, production of urine, to maybe even anuria, where you shut down production of urine altogether. Metabolic acidosis. So your pH is going to go up. Okay, you'll see things like lactic acidosis. We'll be measuring lactic acid levels all the time. Thrombocytopenia, as your platelets come in and they're trying to, they get worn out. Okay, they're an acute phase reactant. Your platelets usually kind of go up in the immediacy of an infection. And as we deplete those platelets, you get thrombocytopenia. And then you can even have what's called obtundation. You start, losing your consciousness. You get hypotensive with endotoxic shock, even in the face of pouring in all kinds of fluid. So we pour in, 9.9% saline or lactate ringer. Then they start pouring in blood products and they'll pour in things like hetastarch. These are things that are trying to keep the fluid in the vascular compartment. Lipopolysaccharide, like I said, is conserved evolutionarily. We see it on the beginnings of some of that archaea bacteria almost to like across all the gram-negative bacteria. There's even some remnants of it in some of the gram-positive bacteria have some lipopolysaccharide-like molecules. Shouldn't almost say that to confuse you. It is deep within the intact cell membrane of the gram-negative bacteria. And we respond to it strongly, all right? Now, when we move from the outer membrane inward with the cell wall still going through, we next have that layer of peptidoglycan, okay, which we know is made up of our repeating units of NAG and acetyluramic acid and NAG. The outer membrane is strongly attached. Like I said here, it's basically almost stapled to that underlying peptidoglycan by a little adjoining LPP protein, all right? That peptidoglycan is running through an area called the periplasm. The periplasm is basically delimited on one side by the outer membrane and on the other side by what we call just kind of this whole cell wall component leading up to the inner membrane, okay? That periplasm is an aqueous compartment. We always have membranes separating our different aqueous compartments, but it's more viscous, thicker than the cytoplasm that's inside the bacterial cell wall proper. And this viscous periplasm allows the gram-negative bacteria to sequester a bunch of its harmful degradative enzymes. And there will be some of those chaperone proteins in there that will help it bring in nutrients and carry it to the inside of the cell membrane eventually. Now we get down to the inner membrane, okay, the plasma membrane. Bacteria lack all of the membrane brown organelles. So all of the membrane kind of associated functions have to be associated with the inner membrane, okay? All the functions like lipid biosynthesis, energy production, secretion and transport of proteins in and out, they exist in bacteria, and all the proteins that pretty much do this are going to be located with the inner membrane, because there is no membrane bound endoplasmic reticulum and Golgi apparatus or any of that stuff. All right, so now that we kind of have the idea of our structure, now we're going to go through in the order that your book does. But I want you to remember, don't waste a whole lot of your time necessarily memorizing what's an alpha proteobacteria from a gamma proteobacteria. You'll get to organize the bacteria in your mind as we go through the systems. So this is going to be an introduction to a lot of them and how they're kind of classed together based on some of their DNA characteristics and structure. But what becomes useful for us medically is to organize things by what infects the skin, what infects the urinary tract, what infects the cardiovascular system, okay? All right, so the alpha proteobacteria, we're talking all about gram negatives. And all the alpha proteobacteria are going to be what we call obligate or facultative intracellular bacteria. Obligate meaning they absolutely have to be inside the cell, or facultative, they prefer to be inside the cell, but they might be able to exist outside of the cell for some bit of time. All right. Most of the alpha proteobacteria are oligotrophs. Whenever you see oligo, means some of the time, right? So these are organisms that are capable of living in low nutrient environments, like deep ocean sediments or the undersurface of the soil, okay? Now, as a class, these bacteria include what we call the protomitochondria. In our endosymbiotic theory, back in evolution, we know that one of our eukaryotic ancestors at some point engulfed a bacteria, and they decided to live harmoniously together, with that bacteria becoming basically the mitochondria and carrying out all the respiratory activities for the cell, making all the oxygen, taking all those reduced coenzymes, stripping them of their electrons, and then adding all that energy into ATP. The alpha proteobacteria, not by chance then, includes bacteria that are able to fix nitrogen, chemoautotrophs, converting decayed matter to chemical energy, and chemoheterotrophs. Now, one of the big bacteria in this class that we want to learn about are the rickettsias. Okay, rickettsias are going to be non-motile, gram-negative bacteria that do not form spores, and they're pleomorphic. So if we know they're pleomorphic, our classic pleomorphic bacteria was our mycoplasma that really didn't have a cell wall to speak of, right? So the rickettsias do have a cell wall, they're gram-negative, but it's like 5 or fewer layers of that, both peptidoglycan and lipopolysaccharide. They're kind of actually cell wall deficient, okay? So they can take on a couple of different shapes. So you might see them forming kind of cocci, coccobacilli, bacilli. They even kind of come in long thready shapes. Rickettsias will always be transmitted to a host, us, through a vector, meaning an arthropod, like a tick, fleas, lice, all right? These are obligate intracellular pathogens. So they have to live inside of another cell. So when they're not inside our cell, they are going to be inside the cell of a tick or a flea or the lice, okay? They require part of their life cycle to occur inside of host cells. So these are bacteria that we really can't grow very readily in culture. We're not going to plate them out on our, gel agar. We're going to have to grow them up inside of other cells. So it's expensive, it's time consuming. It takes a lot of, highly trained technicians to do this. And so we usually either grow them in tissue cultures or embryo cultures. When we say embryo cultures, a lot of times it's an egg. All right, we grow them up in eggs. When they aren't growing inside of cells, they're inactive. Okay, they're only active when they're growing inside of cells. They can't synthesize their own ATP. They need to parasitize the ATP from their host cell. And we'll talk about these a little bit more later on. So when we're talking about rickettsias, the two diseases for rickettsia that we're concerned with are typhus and Rocky Mountain spotted fever, which is actually a misnomer. Rocky Mountain spotted fever is more likely to occur in the Appalachian Mountains than in the Rocky Mountains, but discovered in the Rocky Mountains. So go figure, right? Everything ends up being difficult.
I have a question.
Yep.
So I'm a little confused between like these and the oligotrophs. So like I'm just a little confused. Are these considered one of those, or like the rickettsias?
Well, these aren't really oligotrophs so much. These are really parasites. So they're fastidious organisms. Fastidious, whenever we say fastidious in terms of bacteria, we mean that they have very specific nutrient requirements. They're usually hard to grow up. So they're going to have to be grown up inside of a cell, and they parasitize human cells. Okay. They need to take all of their energy from us. They're not going to be making their own energy. Okay.
Okay.
All right.
And then it is obligate because it needs to be inside the cell.
It needs to be, yeah, exactly that. Yeah. It needs to live inside of a cell. Sometimes we'll find our bacteria, floating around out in the environment, or living in the fluids that are in between our cells, like maybe floating around in your blood or your urine or something like that. These will not. These have to live inside of an actual cell of yours. Okay. They're going to parasitize and make their way inside of your cell. And there they will live and flourish. Okay.
Okay.
Outside of your cell, they're dead. They're either inside of your cell living, or they're inside of the cells of a tick or whatever vector they use. Okay. So like in the case of Rocky Mountain spotted fever that we're going to talk about in a minute, they live inside of the ticks. Okay. And when we get to typhus, they live inside of, the flea that lives on a rat. All right. Okay. So rickettsiae causes Rocky Mountain spotted fever. It's gram negative. Okay. Because we're talking about all this alpha proteobacteria that's all gram negative. And because it has that pleomorphic, it has a kind of deficient cell wall, it's really takes on the form most often as a coxobacillus, somewhere between the rounded cocci and a rod of a bacillus. Obligate intracellular is going to either live in our cells or is going to live in the vector cells, that of the tick in this case. Okay. So the treatment, and I'm never really going to test you on treatment. I'm just telling this, and I put it on the slides now because people are always asking what the treatment is. The treatment is going to be an antibiotic in the tetracycline family, doxycycline, minocycline, tetracycline itself. All right. Signs and symptom when you get Rocky Mountain spotted fever, you start getting a fever and a rash with a history of the tick bite. Okay. And the rash kind of goes in like a bullseye fashion, or we'll have a picture going around it, around the tick bite. It begins on your extremities where the tick bites you, and it kind of moves toward the trunk of your body. And you get bitten and the rash starts developing about 48 hours later. All right. This can be life-threatening. You can get Rocky Mountain spotted fever that can cause such an inflammatory reaction that you end up with inflammation of the meninges, the covering of your brain. Okay. All right. And I know sometimes this is almost like a new method of studying. A lot of it is, yes, drugs and bugs. You got to memorize the bug. You got to memorize if it lives inside of the cell or not. If it's, an obligate intracellular parasite, you have to know, what the course of the disease is. Does it cause a rash? All right. And this is unfortunate, but this is the only way to learn it. And this is what makes micro so hard. Okay. All right. So it's a new way of studying for a lot of people. It's not like there's going to be a lot of analysis that everything is going to suddenly make sense, All right. So Rocky Mountain spotted fever, kind of all this lacy rash, probably bitten by a tick somewhere on their feet or their legs. This is why they always tell you, wear long pants, wear thick socks and that when you go hiking. Here is the critter to beware of. This is the deer tick. Okay, this is going to be the tick that carries the Rickettsia rickettsii inside of it. And when it bites you, that tick, and it goes to suck your blood out, what happens is it injects the bug that's inside of it into you. Okay, so you get basically spread that bacteria into you by an infected tick. And this tick, you'll have a reservoir of ticks that are infected with rickettsia. There will be some ticks out there that aren't infected with rickettsia, but then you get the human being who ends up being bitten by a tick that has rickettsia in its blood, and it starts growing and vegetating. And then the uninfected tick comes along and also bites this person, and then they get infected with rickettsia. So there's always a constant reservoir of ticks infected with rickettsia. Okay, you see how it's kind of a vicious cycle. Tick with rickettsia bites human who doesn't have it. Human then gets rickettsia. Tick without rickettsia bites human, and then tick has rickettsia. Tick goes on to bite another human. It's kind of a vicious cycle.
What happens to the tick? Like when it's harboring that?
It just lives. We call it a reservoir because the tick just lives with the infection in it. Okay. I mean, the tick doesn't really get sick with the bug. The tick maintains the reservoir or the natural population of the rickettsia within its population. That's a good, interesting question. All right. So we have to eradicate the tick from the humans by treating ourselves with the tetracyclines. All right. So when we look at the tick hemolymph cells, here we go, and we can see the rickettsia, these little sort of pleomorphic species growing and flourishing and living inside the cells. Okay. And like I said, it says it's always going to have to require special staining methods because you are going to be looking at, this being infecting inside of a cell. All right, so I can't remember if I put chlamydia in here and your book doesn't put chlamydia in with the alpha proteobacteria. Usually chlamydia is classified in with the alpha proteobacteria. And we will revisit chlamydia a lot when we get to sexually transmitted diseases. We'll also look at it at the end of this set of slides. Chlamydia are also gram negative. they're also obligate intracellular pathogens. And chlamydia causes a series of diseases, okay? And it can manifest itself as a trachoma, an eye disease. It can manifest itself as urethritis, an infection of the urethra, cervicitis, inflammation of the cervix. It can ascend, and this is where it gets really bad, and I know you're anxious to learn it. We'll talk about a lot of it later on. It will ascend up the reproductive tract, particularly in women, and cause pelvic inflammatory disease, which is one of the leading causes of infertility. Okay, so we'll cover this in greater depth later on, but usually classify chlamydia in with the alpha proteobacteria. Another rickettsia, instead of the species here is Prokaryote, named after its discoverer, and this causes epidemic typhus. Again, gram-negative, coxobacillus, kind of a little, cell deficient, cell wall deficient. Again, obligate intracellular, and it likes to live and infect our endothelium, the lining of all of our blood vessels, and ultimately what lines our heart. The vector for this is going to be the lice. I think I said flea earlier. I'm wrong. It's lice. This body louse, okay, the lice, I think it lives in rat populations. And so you see epidemic typhus in times of war because the lice gets transferred very much amongst all those GIs living down in the trenches, not being able to bathe and have proper hygiene. And then there's all the rats running around. And so typhus, you get a high fever, you get myalgias, you get headaches, cough, and you can get a rash that also goes on the trunk instead of like the Rocky Mountain spotted fever that goes from the periphery to the trunk. This one goes from the trunk and spreads outward to the hands and legs. Okay. All right. So both of these, I just thought I'd throw in the term, are called zoonoses, or a zoonosis, singular, zoonoses, plural. Zoonotic diseases. We talked about this a lot over the past 18 months with COVID. And zoonoses are infectious diseases where the pathogen, the infectious agent, like the rickettsia, jumps from its normal species, say the lice or the tick, or, whatever animal it likes to live in, a bat, and it jumps to a new animal and is able to survive in that animal. Endemicity, maintaining endemic, living within a population, remains by transmission from the infected animal person to an uninfected arthropod. Okay, so like I was saying, the uninfected tick population bites a human that happened to be infected by an infected tick, and now those ticks become infected. All right. Usually the infection of the human being is incidental. All right. We're not usually the hosts that it likes to live in forever and ever, not the endemic population like the tick or the flea or the louse or the mosquito, but it serves to jump from and get into the rest of the population of arthropods or bats or whatever it likes to live in by temporarily living in us. Okay. So probably I would say 60 to 70% of all of our infections at some point are zoonoses, rabies, anthrax, COVID, okay? Jumping from one species to another species. And when we talk about zoonotic transmissions, and we keep edging around this, and we will do a transmission lab, we will have things like direct contacts. So this is when you come directly into contact with the infection, like through blood, saliva, feces. So if you go and you decide you're going to lick the bat that started out carrying COVID or something, whatever, the rabid animal scratches you or bites you, that's a direct contact. Some things can transmit by indirect contact. So let's say you have a species of beaver that carries a disease, and that beaver is building, its dams out in the river and kind of, dams everything up, and you have stagnant water, and whatever is infecting that beaver spills out into the water, and then it starts to infect other, incidental hosts, like the human being that decided to go and take a bath in that water. Vector-borne means you're going to be bitten by a mosquito or a tick or a flea, okay? And it's carrying that infection in it. So now when that arthropod bites you, it injects that infection into your blood. Foodborne, this, like salmonella or E. coli. We have the bacteria contaminating our food supply, and then we eat things like undercooked meat, okay? Or, the farm workers who are infected and they, poop out in the field on the head of the lettuce, and you don't sufficiently wash the lettuce when you bring it home from Whole Foods. And then you have, E. coli all over your lettuce, and you eat it and you get contaminated with E. coli and you get sick. We also have waterborne illness, and usually that's water that's become contaminated with some sort of bacteria, like, contaminated with feces, okay? And then you either drink the water or you go swimming in the water, somehow you ingest the water, and then you get ill from that. So these are kind of how we get these zoonotic transmissions. So that's just a little aside as we're going through this alpha proteobacteria. This is a chart from your book that includes a bunch of the species, and this is just like lightly touching on the world of bacteria that are alpha proteobacteria. So our, sort of exemplar that we're going to focus on would be the rickettsia, okay? Because these are going to be ones that commonly will affect human beings in the United States of America. Sometimes Coxiella, you'll see Bartonella, sometimes is like cat scratch fever, things like that. But focus on the rickettsia, because we can't learn everything in a one semester course. And I know that sometimes you get overwhelmed by this, so I'm telling you what to focus on. All right, going from alpha proteobacteria, now we're going to beta proteobacteria. Again, these are gram negative, and there's, again, a huge range of species, 400 species, okay? And so they're going to have a whole range of strategies to survive in varying environments. The one we're going to focus on here will be Neisseria species, which includes Neisseria gonorrhea and Neisseria meningitidis, okay? Neisseria gonorrhea, we will really re-encounter again when we get into sexually transmitted diseases. And Neisseria meningitidis is causative of bacterial meningitis. You're seeing ads all the time on TV now for the newer vaccinations against Neisseria meningitidis. We find that the populations this spreads in will be things like students living in dormitories, or new GI recruits also living in dormitories, okay? Spreads like wildfire. So those are the populations that we're telling and targeting to get the meningitis vaccination. All right, so Neisseria tend to grow as diplococci, Diplococcus Sometimes you almost see them forming tetrads together, but the rule pretty much is growing as diplococci. Here they're growing on what's called chocolate agar, which is agar mixed in with some blood and other nutrients that help these colonies grow up. All right, so Neisseria meningitidis has a capsule. And as you recall, a capsule is a virulence factor. All that polysaccharide that leaks out all around it, okay? When it becomes more organized, it's a capsule. When it's less organized, we just call it glycocalyx or a slime layer. There's also a similar thing in the archaea called an S layer. You might have seen it when you were reading the textbook, okay? But the capsule is the big evader, okay? And this allows it to not be recognized by a lot of our immune system. And then we don't form the antibodies against it if that sugar coating kind of covers over some of its proteins that we would normally recognize as a foreign invading bacteria. Now, we make our vaccines against some of the capsular substances, okay? And that's why we're able to have a vaccine for Neisseria meningitidis. We don't have a vaccine for Neisseria gonorrhoeae because it doesn't make the capsule. As another virulence factor, the Neisseria species often have pili. And remember, the pili will help attach to epithelial cells. And this specifically helps the Neisseria species attach to the cells and tissues that it's invading, like the nice mucous membrane of the vagina in the case of Neisseria gonorrhoeae, or inside the urethra, or inside the anus, or inside the throat. And this is one of those things we'll talk about when we also talk with STIs about sexual behavior. Yes, you know, everybody has sex in different ways, and the mucous membrane of the throat, the anus, the urethra, the vagina, they're all mucous membranes. Remember what mucous membranes are? They're in contact with the outside world, but they're wet linings. They're usually only, you know, a few cells thick. And so that pili helps it adhere to whether it's your throat, your anus, your vagina, or your urethra.
I have a
Also,
question.
yep.
So you said that the capsule virulence factor, but I was just confused because, like, isn't the capsule is what allows the meningitis to have like a vaccine?
Yes, yes. And you'll get to understand this better when we talk about the making of vaccines. So the bacteria will find ways to evade our immune system, okay? So they develop this capsule that covers and hides all of its proteins and things like that. But then, you know, you build, what is it? You build a better mousetrap, you make a smarter mouse. In this case, we become smarter. So what we end up doing is we make our vaccines by, they're called conjugate vaccines. So we attach little pieces of the sugar coating of that bacterial capsule to something larger so that we make an immune response against those capsular sugars.
Okay.
All right. So they do that, we fire back. You know, this is what happens. This is, like I said, it's nuclear war. There's no détente here. They fire, we fire. They fire again, we fire. Eventually they'll find some way of evading us, and we'll have to come back and find some new way to counteract them. Okay. We make antibiotics to kill the bacteria. The bacteria will find things like, literally, you'll see when we get into the antibiotics, they'll put in little pumps that as soon as we're attacking them with antibiotics, they're pumping them right back out, like right back at you, baby, you know? You're going to fire that antibiotic at me, you know, back in your face. It's really amazing the ways the bacteria evolve to counteract everything we throw at them. Okay, the next beta proteobacteria, Bordetella pertussis. So pertussis is your key word here, B. pertussis. So this is responsible for pertussis, also known as whooping cough. All right. And whooping cough, if any of you've had it or you've had a kid who have it, is that big gulp of air sounding that gives it its name. And what happens is this species of bacteria will produce several toxins. All right. So virulence factor here for Bordetella pertussis makes toxins. The toxins here paralyze the cilia on our respiratory tract cells. Remember, you have all those cilia that beat up the mucus you learned about in anatomy so that you can cough and hack out that mucus. Well, suddenly, if this toxin paralyzes the ability of cilia to beat the mucus, that directly damages your respiratory tract and you're not getting any of those bacteria out. So you get a severe cough because you want to get that stuff out, but you can't. All right. All right, so here is your slide from the book of classes of beta proteobacteria. Bordetella, okay, for whooping cough. We'll talk about it more when we get to the respiratory system. Neisseria. So this is going to be our Neisseria meningitidis and Neisseria gonorrhea. Moving on to the big section of gamma proteobacteria. This is a big class of gram negatives, and they are bioluminescent. They're fluorescent, right? They're fluorescent. So this includes Pseudomonas. And if you've worked in a hospital, you get to know Pseudomonas by its sweet smell, grape juice smell, okay? Some say even a floral scent. And it's responsible for lots of infections in the body. But Pseudomonas, we really see as like a secondary superinfection, particularly in patients like the burn unit, okay? Or diabetics, okay? It'll get in their foot ulcers. Or cystic fibrosis patients. They take prophylactic medication to prevent Pseudomonas. So some level of immunocompromise usually. Okay, burns, diabetes, cystic fibrosis.
What about like Stevens-Johnson's?
Stevens-Johnson syndrome?
Yeah.
Where did that come from? Stevens-Johnson's, I suppose it could superinfect. Stevens-Johnson's is like a skin, it's like the only dermatological emergency. And you start exfoliating your skin as a big exfoliative rash, usually in response to taking something like a medication that you've had a reaction to.
Mm.
Did you have Stevens-Johnson's? I hope
No,
not.
I work in a medical ICU. We get a lot of it.
Ah, okay.
That and graft versus host.
Yeah. Oh, yeah. Yeah. Okay. I was like, whoa, Stevens-Johnson's. That's when they talk in medicine, we always talk about when you hear hoofbeats, think horses, not zebras. Think of the usual things being usual. Okay, Stevens-Johnson's is like a really unusual syndrome. So whoosh, go back to my long ago knowledge. I have seen one case of Stevens-Johnson's. So in my lifetime, it's horrible, by the way. And it's the only time we really call in. It's the only time the dermatologists ever come in for an emergency, and usually it gets shunted off to the plastic surgeons because they're already in the hospital. So is that what happens at your ICU, Ellie? Okay.
Sorry. Yeah. No, not really. By that time, we're usually caring for them for other reasons. Like they're usually intubated. We're the medical and respiratory ICU. So.
Okay. All right.
Yeah.
Huh. All right. So these Pseudomonas infections are really difficult to treat. Like I said, gram negatives are always a lot harder to treat and have our antibiotics counter them. They're resistant to many antibiotics, and we definitely are constantly sending off cultures trying to find what on earth we can use to kill these things. They also have a great ability with Pseudomonas to form biofilms, and the biofilms definitely protect them from the antibiotics that we shoot at them. All right, so characteristics of Pseudomonas, also encapsulated. Gram negatives, they're strictly aerobic. They like to live in oxygen, and they're rods. Therefore, they don't tend to be fermentive. They don't go to fermentation, they're aerobic metabolism. They also have flagella, which enhances their motility. They biofluoresce. They produce these fluorescent pigments. So kind of you see them as greenish into yellow, pyoverdin, pyocyanin. And the sweet odor, so if you're getting this up in your ICU, Allie, have you smelt the Pseudomonas and how sweet and fruity it smells when you walk into the room of the patient that has it?
I've actually never encountered Pseudomonas.
Ah, okay. So it smells really sweet and fruity. Grape juice is really like I guess the smell most often people say. You get it. I used to do lots of burn unit patients, and you can really smell it in the burn unit patients. And they like to grow in these moist environments. So it's always like this battle of like you're trying to put all these nice moist wrappings on all the burn patients to help their skin grow back. And then, you're also creating the very environment that the Pseudomonas want to grow in. So it's really a fight.
Wouldn't you do like if you had some sort of like wet and then dry on top, would that stop the Pseudomonas?
Well, and then they go like wet to dry, and then we're constantly skin grafting, and it's a battle. The one case where Pseudomonas infects the non-immune compromised host is hot tub folliculitis. This stuff grows and flourishes in hot tubs. And once you get it infecting your hot tub, it is really, really hard to get it out of there. So in those jets, you can see like almost like a biofilm or tape growing of like this kind of grayish black. And if under the right light, it will fluoresce of Pseudomonas. You get a whole biofilm clogging up those jets. You can fight it and fight it and fight it with, trying to Clorox the heck out of it. But sometimes it is a losing battle, and you need to get new airlines and jets put in your hot tub if you get that. All right. Gamma proteobacteria is our next order. So, our next phylum. Now we're getting down to our order of Vibrionales. So our exemplar for this is Vibrio cholerae. What causes cholera? So this bacteria is highly motile. It has a flagella. It is sort of the exemplar bacteria we always used for what is comma shaped. And actually, when we went in, back in I think it was the 1850s into the early century, somebody wanted to call it like Bacillus comma or something like that. So it went through some early name changes, but it's now set as being Vibrio cholerae. Comma shaped, it's a rod, it's aquatic, it lives in pools of water. And as you know from reading your textbook, and you read the story of John Snow and how he discovered the Vibrio cholerae infecting the well in the town. And so cholera spread into the local town, traced it back to the well, and nobody, really wanted to believe him at the time. It tends to also thrive in highly alkaline water pools like shallow lagoon areas. And its virulence factor is it too secretes, causes a hypersecretion because of a toxin that the Vibrio cholerae make. Okay. And this toxin attacks your intestinal epithelial cells, which leads to profuse watery diarrhea that is called rice water stool. You have like pale little chunky bits of stool. The genes for the toxin are not actually in the genome of the Vibrio cholerae themselves. They're brought in by a phage, by a virus that attacks the bacteria, but it often gets incorporated into the genome. All right. And there is now a vaccine available for people who live in endemic areas or travel to areas like you're in the Peace Corps, where there's going to be endemic cholera. All right. I wants to skip ahead to another gamma proteobacteria is Legionella pneumophila. Pneumophila tells you it's going to attack our lungs. Legionella comes from the Legionnaires, where we first discovered this back in Philadelphia and infected the air conditioning units at a Legionnaires conference. All right. So this is what causes Legionnaire's disease, also known as Pontiac fever. These critters are thin gram-negative bacilli, again, somewhat pleomorphic, so they have an incomplete or cell wall deficiency a little bit. They inhabit pools of water, but usually it's pools of water that are in things like air conditioning units in bacteria. So that, air conditioning units in like hotels and conference areas, large buildings. So they somewhat get aerosolized. So it spreads in aerosols throughout the building because of being in the air conditioning unit. This, got its cooling water running through it. And then we inhale it and it gets into us and we get Legionnaire's disease, which is a type of pneumonia. And we'll discuss this a little bit more when we get into respiratory diseases. Like I said, all this is kind of a preview of attractions to come. So here you see Legionella pneumophila, and here is the air conditioning unit where they have the water systems that get infected and then it gets aerosolized. Then we have a large family of enteric, meaning in your intestinal tract, bacteria that are amongst the gamma proteobacteria. And these are facultative anaerobes, meaning they prefer an anaerobic environment, but they can grow in oxygen, right? They also ferment carbohydrates. And we're going to talk about two distinct categories of enteric gamma proteobacteria. The coliforms, liking to live in your colon, and the non-coliforms. The coliform prototype is Escherichia coli, E. coli. This is our classic gram-negative rod that does not form spores. Okay, let's repeat this because we'll have a question coming up that you might encounter about gram-negative rods and those which form spores and those which don't. This one does not form spores. It can be either motile or non-motile. So these will tend to ferment lactose completely and produce acid or gas. Now, they used to always be called lactose fermenters, but because they sometimes don't ferment lactose, but they'll ferment some other sugars, it's more clearly defined now by these contain an enzyme known as beta galactosidase. All right. I'm telling you all this, but we're probably not going to get down to that level. All right. The non-coliform types of bacteria are ones that don't ferment these sugars, particularly lactose, or do so incompletely. Okay, so these are the big difference between the coliforms and the non-coliforms, what's going on in your intestine. And so these will contain all these species that you've heard of, probably, like Salmonella, Shigella, Yersinia pestis, which is the cause of plague. All right. So Shigella and Salmonella both are, diarrhea causing agents.
But this might seem like a silly question, but if you got infected with E. coli and you had that beta galactose, if you didn't eat any lactose, would you even have symptoms?
Oh yeah, because it's not the only thing that it does. It's just saying that it can ferment it. It doesn't live off of lactose alone. Okay. It will live off of all kinds of nutrients that you have in your intestinal tract. But this is how we identify it in lab, is we'll put it in a test tube with lactose and see if it ferments or we cause gas to bubble up in the tube versus the species that don't. Okay. So this isn't about the bacterial survival, it's about our identification. So this is actually a really good question, Allie. All right. Okay, so talking about E. coli. So if we just want to bring ourselves back around, when we're looking at all the taxonomy of all this, we classify it under the kingdom of eubacteria, true bacteria, in the domain of bacteria. It's part of Proteobacteria, that phylum. And then we break it down in the class of Gamma Proteobacteria. It's in the order of Enterobacteriales and in the family of Enterobacteriaceae, and its genus is Escherichia, and coli becomes its specific epithet or species level. All right. You obviously don't need to memorize this, but every now and then we get asked about kind of bringing us around because we get lost in all of what is this Gamma Proteobacteria. If we're going to be true microbiologists and you really want to go on in microbiology, you would start to get to know all your taxonomy very specifically. Again, we're a little bit faster and looser with medically oriented microbiology because we're going to go so much more on how we're infected. Okay, what systems are infected. I'm just checking the time, 6:37. All right, so E. coli, gram-negative rods, they're going to possess a lot of adhesive fimbriae, so they can adhere to our intestinal tract. Even when there's a lot of stuff flowing through it, they can still adhere to our tract. They're going to have a cell wall, and that's going to have that outer membrane that contains the lipopolysaccharide because it's gram-negative, the periplasmic space with that peptidoglycan layer, and then it's going to have the inner cell membrane, just to bring us back around. Some strains of E. coli will be pileated, have lots of pili on them, and some of these pili will allow it to behave as reproductive pili. Am I seeing somebody's little kitty cat whiskers come on? I think I just saw a kitty cat out of the corner of my eye. Some of the so the reproductive pili will.
Yes, sorry, that's my cat, Wolf, or
hey,
one of them.
We gotta love the cats. I love all the animals. Cats. I was a zoology major before I went to med school, so I like seeing everybody's pets. And as you know, you all see my fat cat when she decides to prance in around here. So the reproductive pili can transfer plasmids from one bacteria to another that will confer quite often survival or evolutionarily beneficial qualities from one E. coli to another, or maybe from one E. coli to some other species. E. coli itself only has its circular chromosome, and sometimes it also contains a plasmid in there, okay, which has some extra chromosomal DNA. So E. coli probably has the chromosome that is the DNA that is best known to all lab workers everywhere. It's been, the generalized chromosome has been completely sequenced. All right, so it was first described by Escherichia, and it exists in our colon, living in a mutualistic relationship. Most of the E. coli in our intestines are not harmful to us. And you'll see when we get to studying this a little bit more, when we get to the digestive system, we're going to classify E. coli by type into 6 different categories. So we're going to have different strains of E. coli. We'll call it like enterohemorrhagic E. coli. That's going to be like our 0157 E. coli that causes big, bad, bloody diarrhea. Okay. Then we'll have like a kind that might cause traveler's diarrhea. So we'll classify all these later on. We'll see that some strains of E. coli carry a deadly toxin, also the same toxin that's in Shigella, so it's called Shiga toxin. This is going to be one of our most potent bacterial toxins that we know. So this toxin would be a virulence factor. When it enters human cells, the Shiga toxin is going to interact with our ribosomes and stop protein synthesis. This is what causes it to be so destructive. Lack of protein synthesis will lead to cell death and hemorrhagic colitis. So then you'll see inflammation of the intestinal tract, bloody diarrhea, and we can go on to develop what we will call later on a hemolytic uremic syndrome. So it can later on affect your kidneys as a sequelae, okay? Other strains of E. coli can cause less harm, but still cause harm. Like I said, traveler's diarrhea. You have it for a little bit, you know, you went on a trip, you picked up a new strain of E. coli, and this tends to be more of a watery diarrhea. It doesn't tend to be a bloody diarrhea. All right, this stuff that looks like ground hamburger is really just a bunch of salmonella, okay? Salmonella typhi, which causes typhoid fever. And that's a type of gamma proteobacteria. All right, so pathogenic salmonella, let's go back to salmonella species. These are also a type of gamma proteobacteria, and these are classified under Enterobacteriaceae. And these are gram negative, and they're flagellated, they're anaerobic, okay, so they don't like oxygen, and these will possess 3 major antigens by which we will type them. An H antigen, which is the flagellar protein, an O antigen, which is a somatic or body antigen, and what's called the Vi protein. And this is not always in every strain of salmonella. So we'll decide whether it possesses it or whether it doesn't possess it, and then type it by that. As a facultative anaerobe, it makes ATP by aerobic respiration, okay, if oxygen is present, but it can switch to fermentation and just ferment sugars going down that glycolysis pathway, okay, and not going through the Krebs cycle in oxidative respiration. Salmonellosis, as you know, is a bad gastroenteritis, okay? Along with the gastroenteritis, you usually get fever, you can get septic infections, and you can even have an asymptomatic carrier state, all right? Some types will cause inflammation of both the small intestine and the large intestine, you know, and with that, you can get all those gastroenteritis symptoms, nausea, vomiting, fever, all right? The type or serovar, Salmonella typhi, causes typhoid fever, not typhus, typhoid fever, with symptoms that include fever, abdominal pain, and the skin rashes. Now, if it's ingested in food and it can then survive the harsh acidic environment of the stomach, the gastric acid, to invade our mucosa, the lining cells of the small and large intestine. All right. When it invades the cells, it's going to cause that great inflammation, okay, in which we're going to remember from anatomy, those of you, cytokines. We're going to release all these cytokines that recruit and call all these inflammatory cells that will cause our local inflammation that will lead to the ulceration and destruction of the mucosa, as well as all the symptoms of the diarrhea. All right. Now, the salmonella that causes typhoid fever and enteric fevers will spread mainly from person to person via what we call the fecal oral route. So what's fecal oral transmission? It means somebody's poop has likely gotten somewhere that makes it into your mouth, like on somebody's hands, like the person preparing your food at a restaurant ran to the bathroom and ran back in the kitchen and prepared your foods. Or like I said, the worker out in the field who poops out in the field, it gets on the head of lettuce or your fruit or something, and it's insufficiently washed when it comes back from the grocery store. With salmonella, we also have a pool of asymptomatic carriers. They don't have that inflammatory reaction. They don't get the diarrhea. They don't get sick. And this is what happened with Mary Malone, Typhoid Mary, back around the turn of the century. She was a cook in a number of homes in New York. The people that she cooked for all got sick. The public health people came and traced it back to her, discovered that she was the cause, and they quarantined her. They took her off of quarantine, told her not to be a cook anymore. She went out, got another job as like a laundress, didn't make as much money. So she snuck back into cooking, cooked for more people, got people sick, ran away from all the public health workers that were trying to find her, actually went and got a job cooking at Sloan Memorial Hospital in New York City. And then they had an outbreak of epidemic typhus through the hospital. They traced it back to her again, but she continued to kind of like outrun the authorities. They literally had to get a physician named Josephine Baker to sit on top of her in an ambulance and hold her down. And I think for the rest of her life, if I'm not mistaken, they actually quarantined her for the last 10 or 20 years of her life, like in a quarantine prison, because they could not stop her from cooking for people. So fecal oral route, Typhoid Mary, remember the story. Hence, today we have all the signs to wash your hands, right? After working in a restaurant, after you go to the bathroom. All right, so there's salmonella. All right, so gamma proteobacteria, here's your list. I would say kind of pay attention to the Enterobacter, the Escherichia coli. We'll encounter some of these later on. We'll encounter Klebsiella when we get into pneumonia. Remember the Legionella, remember Pseudomonas that we've talked about, the Vibrio cholerae. We'll get later on into Shigella and all that. Probably not as important to remember it right now. All right. So then we have this class, this small class of gram-negative proteobacteria. And there's very few of these that are pathogenic. So, so you can kind of somewhat not pay so much attention to the delta proteobacteria, but I keep it in here for some of those of you who are going onto oral hygiene school, because periodontal disease, there is one of these small bacteria called Desulfo Vibrio orale, and it causes some of the periodontal disease in the gums. We'll spend some time on strep mutans, which also is one of the causes of cavities. All right. All right, so I didn't highlight any of these. In the Epsilon proteobacteria, these are also gram-negative, of course, and they're microaerophilic. So they only require a little bit of oxygen to survive. And we have two clinically important genera. the Campylobacter, and the Helicobacter. So Campylobacter species, again, they're curved little motile rods, often flagellated. And so these tend to cause severe enteritis, inflammation in our small intestine. And it's been under-recognized, and now the CDC is saying they think this is problematic for food poisoning at more than 3 times the rate of salmonella. So there's a lot more attention being paid to Campylobacter. So it's considered the major cause, the most common cause of bacterial diarrheal illness. The species that you'll always hear about is C. jejuni. This one is what's common in developed countries due to contaminated poultry. And chickens often harbor C. jejuni in their gastrointestinal tract. And as you can imagine, most of this gets transferred in the process of butchering. You cut up the animal and the intestinal tract gets split open, and the bacteria spreads from there to other parts of the animal, which we then consume. Whoops, skipped again. All right, Helicobacter. This is what you think of as causing duodenal ulcers, gastric ulcers. So Helicobacter pylori, there will be quite a number of questions on your first PBL about Helicobacter. Some of it is the material you need to answer the question is actually in the stem of the question itself. Okay, so it's trying to see if you can think about bacteria in those questions. All right, so somewhat it was a beneficial member of the stomach microbiota, but then there is a Australian researcher who found that it actually is the most common cause of chronic gastritis and ulcers in the stomach and the duodenum, the early part of your small intestine. Chronic infection with Helicobacter then leads to stomach cancer. Helicobacter has an amazing ability to live and thrive and survive in the acid environment of the stomach. Now, the acidic environment itself really isn't suitable for its growth. But what Helicobacter does is it produces enzymes like urease that modifies its environment, makes its environment more alkaline, neutralizes locally the stomach acids so it can grow. When it does this, it produces ammonia as a result of the urease, neutralizing the stomach acid and increasing the pH, neutralizing, making it more alkaline. We can measure, there's a urease breath test that we can use to measure the presence of this Helicobacter. The flagella on this bacteria actually plays a big role in its ability to colonize the mucosa. So here you see H. pylori, you see a bunch of its flagella. Oh, and by the way, there was a flagella labeled, and I don't know what all went on with that weird day of the blackboard, but I corrected the answer. And most of your answers on at least, I think the first 3 homeworks should now be all corrected. I don't know if you saw that updated, but I spent a bunch of time grading the other day. All right, so Helicobacter leads to ulcers, which leads to chronic ulceration, which then can lead to stomach and duodenal cancer. All right, Campylobacter and Helicobacter, both gram-negative, both kind of spiral, comma to spiral shaped, and both causing varieties of enteritis. All right, so then we have 4 classes of gram-negatives that are not considered part of proteobacteria. And like I said, chlamydia most often is classified with alpha proteobacteria. So we somewhat talked about it there, but we'll talk about it some more now and again, also with our sexually transmitted diseases. Then we also have the spirochetes, what we call the CFB group, and then the planktomycetes. All right, so let's see where we are on time. 6:54. Okay, so it's pretty much time to stop. So I guess maybe we'll pick up with chlamydia next time. I was gonna open the PBL tomorrow, but I think I'll open it up after we finish this up on Thursday and then keep it open probably till Wednesday of next week. Do we have more questions, or are you just spinning your head full of bacteria right now? Spinning your head full
I
of
have a
bacteria.
question.
Go ahead.
So why are all of the protobacteria gram negative? Is there a special reason behind that? I guess.
Because it's a classification of gram-negative bacteria.
Oh, it's just because of that. Oh, okay.
Yeah.
It doesn't have anything to do with like the bilipid layer or anything.
I didn't hear quite what you were saying.
Also, it doesn't have anything to do like with the bilipid layer at all, or it's
Well,
just because that's how it-
proteobacteria is a phylum of gram-negative bacteria that we then divide into alpha proteobacteria, beta proteobacteria, gamma proteobacteria. So these are all gram-negatives. It's just the phylum, it's just the subclass. We haven't even started talking specifically about any of the gram-positives yet. Okay?
Yeah. And then I also had another question.
I know the taxonomy is confusing.
Yeah. I also have one more question with the pseudomonas.
Pseudomonas? Yep.
Yeah. So would that be considered was it faculative or folliculative type of
Is it a what? I can't hear you.
it's I forgot what it's called, like it's obligate and there was the other one.
Oh, Pseudomonas is not obligate intercellular.
So it's the other one.
Obligate intracellular would be like the rickettsia and the chlamydia.
So then
I
that
think
would
she's asking if it's facultative.
facultative, yeah.
Pseudomonas likes to grow in oxygen. Okay. It's I would say it's an obligate, tends to be an obligate aerobe. Does that help you?
Yeah, kind of.
It's strictly aerobic. It likes to grow in the presence of oxygen. Okay. We're not going to grow it up like we have what we call deep slants, where we grow bacteria and we hide it from the presence of oxygen. So something that is an anaerobic bacteria, like Bacteroides, we're going to grow it in a non-aerobic, lacking the presence of oxygen. Pseudomonas is strictly aerobic. So we're going to always grow it up where there is oxygen. So you don't tend to find Pseudomonas growing like in the deeper tissues. It likes to invade like the infections, like in burns that are still somewhat open to the oxygenated environment. Is that making any sense?
Yeah, so then when would it be considered facultative? Like when will something be considered that, like facultative?
When is something considered facultative?
Yeah, facultative, yeah.
So facultatively anaerobic?
Yeah, or just that in general, yeah.
Okay, so a facultative organism prefers to grow by aerobic respiration, but it can switch to fermentation. So when you're facultatively aerobic, you prefer to undergo from glycolysis through your Krebs cycle and then use mitochondria to make all the ATP. But remember, like in our cells, if we don't have the mitochondria and that available, we can't go through the Krebs cycle. We keep going through glycolysis and making just 2 ATP. We're just netting 2 ATP for every sugar that we burn. Similarly with bacteria, if they have oxygen available, they will use oxygen to respire. But some bacteria will switch and are able to switch to surviving through fermentation. Okay, in that case, they're facultatively aerobic, prefer to operate under oxygen, but can switch to fermentation. I should probably, I'll talk about the oxygen environment that we grow bugs in. See, this is usually what happens in lab, that we do all this stuff in the lab. But I'll spend some time next time we meet in clarifying that. And I'll show you kind of, if I show you the levels of oxygens that we grow bacteria in different tubes, maybe it'll make more sense what's aerobic, what's faculty anaerobic, and what is completely anaerobic, and try to clarify that for you, because these are really good questions. Okay.
Yeah, okay, thank you.
Yep. Yep. I could probably even bring up a slide or an image right now, just while we have the question. I'll show you a little bit. Let me see if I can bring up something. Because I used to, in my old class, teach on this just so that we don't go unanswered. Oh boy, this is really tiny. Well, I'll try to blow it up. Hang on one second. All right. All right, let's see if this helps. Okay, so this will be talking about the presence of oxygen and the growth of various microbes. So obligate aerobes mean that the bacteria is only going to grow in the presence of oxygen. Can you see this, by the way, that I brought up?
Yes. Yes.
Okay, so when we put it in growth media, you're going to see all the bacteria is going to grow where we have oxygen at the top of this test tube. All right, so it's going to cluster here. Down as we go farther in and there's no oxygen in the test tube, it's not going to grow because it has no oxygen down there that it can get to. Facultative anaerobes prefer to grow in oxygen, but like I said, they can grow by fermentation, not in the presence of oxygen. So you're going to see a cluster of them where they prefer to grow in oxygen, but they'll grow throughout the whole test tube. Something that's an obligate anaerobe is only going to grow where oxygen is not. So up here at the top of the test tube where we have all this oxygen, nothing's growing. But deep down in where there's no oxygen, you find it'll grow. What we call aerotolerant anaerobes are kind of a lot like facultative anaerobes. They grow anaerobically, but they still can continue to grow in the presence of oxygen, meaning oxygen's not going to kill them. All right. And then we have what are called microaerophiles. Here, they prefer to grow in a low oxygen environment. So they don't like to grow up here where oxygen is high. They also don't want to grow down in the anaerobic area. So they kind of fill out in the middle area where the oxygen tension is like lower than what's present in air, lower than atmospheric oxygen. Okay. So this is what we're talking about by what's being anaerobic or what's aerobic. So the bugs that live on top of your skin, by and large, are probably going to be aerobic because they're in contact with oxygen. Things that grow like deep down inside of your gut, like where there is not an opening to get air in, probably are going to be things that are anaerobic. They're going, there's no oxygen in the spaces in your gut that is not open to the outside world, right? So we'll discuss that when we get into, say, like Bacteroides and some other of the anaerobes that like to grow in the deep tissues of our body where there's little to no oxygen. Okay, does that help?
That was fantastic. Can we have that print off or someplace posted on Blackboard?
I'll load this up tonight onto Blackboard along with the slides that we just discussed. Okay.
Thank you.
Yep. All right. And I will see you on Thursday, and we'll kind of review this for anybody who dropped off and didn't stay for these last few minutes. Okay.
Could I
All right.
ask one quick
One more
question?
thing to say.
Yeah, go ahead.
It's just I was trying to look at the video from last Tuesday, and it has the audio, but there's no like actual video. There's nothing to see.
There was no video?
No, it just had the audio.
Huh. Okay, I'll try to load it up again. I don't know why that is. We've just had crazy problems with Blackboard lately. I'll reload it up. Okay. Anybody else?
I had
Yeah,
a question.
You
Go
can
ahead.
go. You're fine. I'll go after you.
The lab from last week is due before the lab from this or lecture on Thursday, right? Correct?
I believe so. Yes. I try to make it due
Okay.
at 5 o'clock before the next lab.
Okay,
Do you
right.
guys need more
Thank you.
time? Do you want more time? Okay.
No, I just couldn't remember. I'm
Okay.
good though.
Okay. I'm always going to try to make lab due 5 o'clock the day of the next lab. So we'll be consistent. Okay. Anybody else?
Yeah, I had a question about homework 4. So I don't know. I think I'm like overcomplicating it, but I know you said that the question about the anaerobic gram-negative bacilli can't be E. coli, but can you explain to me like why it doesn't fit that question? I guess I'm just like confused.
Okay.
on it.
I can't remember what the question is off the top of my head.
I think I have it up. I can read it. It says anaerobic gram-negative bacilli are common microbes that populate the intestinal tract. Name a bacterium that we discussed in lecture that fits this description.
Okay. Anaerobic, gram-negative bacilli. Okay, so it's anaerobic and it's a rod, right?
Yeah.
That we talked about. So what do we know about E. coli?
It's gram-negative and it's bacillus shaped, right?
Okay, so you know it's a gram-negative rod. Do you know anything about whether it is aerobic or anaerobic?
It says aerobic but facilitative. Is that why it doesn't fit it? Because it's not fully?
It's not anaerobic, is it?
Oh, it's not?
I don't know. What do you tell me? What do you think?
I thought it was like the
I
one
do know,
where,
but I mean,
yeah,
what did we learn about E. coli today?
I thought it was like the where it could grow in either, but it preferred the oxygen, wasn't it? So that's
Right.
why it doesn't.
Right. So that means it's not anaerobic,
Oh, okay.
facultatively anaerobic. So if we go back to our share right here, facultatively anaerobic is very different than anaerobic, obligate anaerobic. Maybe I should put the word obligate in there, but an anaerobe. And I don't think we've gotten to the point yet where we've discussed the actual anaerobe.
Okay, we haven't. That's why I was confused because I was sitting here and I was like, I thought we were going to go through lecture today about this one. So I was thinking about it the whole time.
You
like,
were zeroed in.
I was like,
Targeted
it can't
for
be
that.
salmonella because that's facilitative anaerobic too. So I was like, I don't know.
Right. So yeah, so it's a good thing we're having this little discussion here on anaerobic and aerobic and all that. Yes. So it's a good reminder to go over this again. So we'll start next lecture going over this. And I will actually put a note. I think I've put a note before, being very careful for people to answer that question, because I know last year a lot of people picked the wrong one. And what I'm trying to do, and why I'm not telling you the answer, is part of the deficiency of teaching online is that you guys don't get the opportunity to go through lab and test and run things and see like, oh, is this aerobic? Is this anaerobic? Is this fermenting sugars? Is this not fermenting sugars? Is this growing in yellow colonies or is this growing in white colonies? And normally, you use known bacteria to learn how to do the tests. And then at the end of the semester, I give you a test tube of unknown bacteria, and you have to decide what order you're going to run the tests. And it's not as simple as saying, oh, it's gram positive, it must be staphylococcus. There's a world of gram positive bacteria. Okay. Just like, oh, it's a gram negative rod, it's got to be E. coli. That's kind of like our stereotypical gram negative rod. But there's lots of things that are gram negative rods other than E. coli, and you have to run tests. And so one of the tests you could run is to see, does it grow aerobically or anaerobically? And if I'm telling you it's anaerobically, it can't be E. coli, because E. coli is facultatively anaerobic. So it will grow in oxygen or without oxygen. Okay.
Yeah, thank you. That made a lot of sense.
The bug I'm trying to lead you to is completely anaerobic, hates the presence of oxygen. Okay. So that's a big difference.
And then,
and that's what that whole question is trying to get you to do, is try to think down all the possible pathways of how do I differentiate 1 gram negative rod from another gram negative rod.
okay. And then homework 4 is due tomorrow. Is there any way that you could extend it till like Thursday, just so we can go over that?
Exactly. Yeah.
Thank you so
I'll
much.
do that. So, okay. Any other questions?
I did. I had one for the lab. I'm so sorry. I'm sure
okay.
I'm overthinking this.
All right.
So
Don't worry.
It's number 24, and it's talking about the inoculating of the loop with the flame in each turn of the quadrant. And it's a true or false. That's true until you get to quadrant 4, where in between 3 and 4, you don't inoculate and put it in the flame. So it'd be false, right?
Right. You don't inoculate between 3 and 4.
Okay. I was like, I'm way overthinking that, because it originally you want to say it's true because you are until you get to 3 and 4. Between 3 and 4, you're not, because you want to colonize.
Right.
Right.
Exactly. And I'll tell you kind of the truth of the matter is, people could argue with me. There are some, everybody gets into their different little lab techniques. Now, the real lab technique when you're trained is not to flame between quadrants 3 and 4. There are some people that do it, but I would say that's poor form. Okay. So, so how you should be trained is not to. Now, I can't remember what the question says offhand, but if you get it marked wrong, and if it's for that reason, argue it with me, and I'd give you the points. Okay.
Okay. I got that question wrong. Actually, I picked false, and it said it was not false.
Okay, so I can't remember what the question is. It might be false for some other reason.
It's saying, I have it up. It's saying the inoculating loop is flamed at each turn of the quadrants when preparing a strike plate. You're flaming it for 1, then you're flaming it after 1, between 1 and 2, and 2 to 3, but you're not flaming it from when you're going from 3 to 4 quadrant.
Right. Right.
So it should be false, but
it should be false. And if it's not false, what I'll do is I'll go back in, because I haven't graded that one yet, the streak plate. I will catch it, and I'll switch the Blackboard answer to make sure that it's false. So it'll automatically regrade everybody who had it if they got it wrong.
okay, like I said, I just wanted to make sure, because like I said, it's true up until that point, but I was just making sure I wasn't overthinking it.
Okay. Yep. I always catch answers on regrades, even though most of these are questions I've used before. Like I said, something strange happened with Blackboard. It completely knocked off edits that I made. I had marked flagella. I was careful to make sure the flagella was marked, and then it switched it to the pili. I don't know why it did that. So, but I catch it when I go through and I look over all the papers when I grade them. So. Okay.
Thank you.
Yep.
Thank you so much.
Night.
You too. Everybody have a good night, and I will see you Thursday, 5:30.
Yeah.
And I will make a note to post this right now. I will actually just post this and go change that answer. Okay? All right.
Thank you.
Bye-bye.